Okay, good morning 12th class. Then today we'll discuss about purification of colloidal solution. Okay, actually whatever lyophobic sol will get after different method, either it's a Bredick arc method, okay, maybe it's a dispersion method, it's a peptization process, we have so many methods. Okay, generally they contain some impurities. Okay, so there is a need for the purification of colloidal solution. उनकी ज़रूरत है, otherwise some impurities are present in it. Okay, so to remove that impurities, we'll use some method for the purification of colloidal solution. So that one is basically we have three main methods for purification, and you all are familiar with this also. Now one is, first method is dialysis. Okay, very important. This term I think you have heard in that kidney के लिए जब हम use करते हैं, that one is dialysis. Okay, तो वहाँ use कर रहे हैं, तो I think you are familiar with this also. So again I'll explain this thing. Okay, not in that detail because that one is in bio. Okay, we have to learn only the principle of dialysis. Now next thing is, that one is ultra filtration and another one is ultra centrifugation. Now in case of first one, that one is regarding to dialysis. Okay, what is dialysis? होता क्या है ये? Okay, simple. In case of dialysis, actually it's a process of separating, because purification is there. तो उसके according हम इसकी क्या करेंगे, definition लिख रहे हैं. Okay, now just see. Simple dialysis means it's a process of separation of what, that one is colloidal particles from, okay, those of crystallites, okay, by means of some suitable membrane or by the process of diffusion. Okay, that one is known as dialysis. Okay, just see now 12th class. In case of dialysis, the definition is clear to you. It's simple. It's a process of separating what, that one is colloidal particles या colloid particles from those of crystallite by means of diffusion through a suitable membrane. That one is known as, okay, that is dialysis. Now what's the principle of this one? होता क्या है? तो just see, there is a simple diagrammatic representation. Now this one is a setup. Okay, so the principle is based on the fact that colloidal particles cannot pass through, okay, cannot pass through a parchment or cellophane membrane, while ions of electrolyte, means those who are crystallite जिसका size कैसा है, छोटा है, okay, they can pass through it. Okay, so the solution, the colloidal solution is taken in a bag. Just see, this one is a parchment membrane, okay, and this one you can take that colloidal solution जो भी हमने prepare किया है, that one you can take in a bag. This one is simple, इसको hang कर रखा है. Okay, now this container, this bag is suspended in fresh water. Okay, the impurities, now just see here, this side it's a water, this one is water plus crystallite, this one is a source कि यहाँ से आपको क्या वो supply करना है, water you have to supply. Now this side it's a water plus crystallite. Now in this case the bag is suspended in fresh water. The impurities, okay, slowly diffuses out of the bag. वो धीरे धीरे क्या हो जाएंगी, diffuse out हो रही हैं, okay, leaving behind pure colloidal particles in the solution. Okay, now simple, the water is changed frequently. This one is very important. You have to change water frequently, otherwise what happen, that one is the start diffusing back. जो crystallite वाले particles हैं, they will start diffusing back. वो दोबारा क्या होने लग जाएंगे, अंदर वापस जाने लग जाएंगे. That's why you have to replace the water frequently. Okay, and this is basically a process of dialysis. Simple, parchment bag में हमने क्या ले लिया, colloidal solution we have taken. There is a flow of water. Okay, and simple, in this principle is based that crystallite that particles, colloidal particles cannot pass through this membrane. ये बाहर नहीं आ सकते, but crystallite particles can move, okay, pass through this membrane, so they will move along with water, okay, and you will get pure colloidal solution. Okay, now but in this one 12th class, this process is very slow. ये आपका very तो मतलब this process is actually a slow process. Okay, और हमें क्या चाहिए होता है, चीज़ें फटाफट चाहिए होती हैं, फास्ट, इतना टाइम नहीं है कि हम इतना वेट कर सकें. Okay, तो simple, to increase the speed of this dialysis process, okay, we use electrodialysis. Now in case of electrodialysis, other thing is same, principle is same, only we will put 2 electrodes. Now just see, 2 electrode means we will pass electricity, and just because of that the speed increases and you will get colloidal solution. So simple, the ordinary process of dialysis is slow. So to increase the process of purification, that dialysis is carried out by applying electric field. So this process is known as electrodialysis. Okay, this one is only for increasing the speed of purification. Okay. Okay, 12th class. Now one dialysis, hope so it's clear to you, and electrodialysis also. Now second method is, that one is ultrafiltration. This one is also a slow process, and generally to increase this process we will apply some pressure or some suction is required. Now what happen in case of ultrafiltration? I think you know about filtration. Simple thing, filtration, चाय हम लोग रोज छानते हैं. That one is tea leaves. You will separate tea leaves from tea. आराम से separate कर रहे हैं. That one is a simple process of filtration. But in case of colloidal solution, because in case of tea leaf the size is large comparatively, but in case of colloidal solution the size is smaller one. Okay, छोटा होता है काफ़ी comparatively. तो instead of filtration process we will use ultrafiltration. So in case of ultrafiltration, generally it's a process of separating colloidal particles from impurities by passing it through graded filter paper. That one is through graded filter paper. Okay, now basically in case of graded filter paper the pore size is comparatively smaller as comparison to normal one. Okay, and generally that graded filter papers, how to prepare it, it's a long process. Okay, process is not required but थोड़ा सा पता होना चाहिए. Okay, कि generally normal filter paper, that one is soaked in colloidal solution, and after that we can dip it to make it more harder. Okay, in formaldehyde solution. तो वो कैसा है, that one is different thing, but you will get graded filter paper. So simple, this process is used for the separation of colloidal particles from impurities. Okay, and pore size is small, so you can get colloidal solution, and that one will allow small particles to pass through it. Okay, so that one is simple ultrafiltration. And to increase the process, you know, pressure or suction is required, otherwise it's a slow process. Now third one, that one we have centrifugation, ultracentrifugation process. Okay, थोड़ा सा कुछ 9th class का याद करो. Centrifugation process, that simple, you can take colloidal solution into what, that one is in a test tube or in a tube, and then keep it in ultra centrifuge and rotate it with very high speed, because their principle is based on the rotation. So when you rotate it with very high speed, okay, so then colloidal solution, colloidal particles get settled down, and after that you can regenerate it by adding some suitable substance. उसमें कुछ suitable chemicals वगैरह add करो तो you will regenerate it. तो simple ultracentrifugation means you can take that colloidal particles in it, जो भी आपका solution है colloidal solution, then you can what, that one is put it into ultracentrifuge, rotate it with very high speed, and in that case particles, colloidal particles get settled down, and you can regenerate it also. Okay, so by this you can get pure colloidal solution or pure colloidal particles. So these three methods, okay, for the purification of colloidal solution. Okay, thank you.